speaking to us about coastal microbial responses to different stressors. And Dr. Sorry, Amelia, were you trying to? Wasn't sure. Okay. So Dr. Bulseco received her PhD at the University of Northeastern in 2018, where she was studying how nitrates affected microbial decomposition and the implications for that on carbon storage. She then proceeded to do her postdoc at the Marine Biological Laboratory in Woods Hole and is now an assistant professor at Eckerd College. She just finished her first semester there, so congratulations on that, especially during a pandemic. That's very impressive. So without further ado, I'll hand it over to Dr. Bulseco so we can learn more about the relationship of microbial ecology and biogeochemistry.
Great, thank you so much. All right, so can I get a confirmation that that's showing up okay?
Yes, I can see your screen. And then do you know how to use the laser pointer on, yep, cool.
Got it.
In case you want it, okay.
All right, awesome. Well, thanks. Yeah, thanks so much for that introduction, Natalie. So first off, thank you all for being here and for inviting me to USF. I really wish we could be interacting in person, but I'm super excited to share some of my research today, most of which took place in Massachusetts during my PhD at Northeastern. So today I'm going to talk about the coastal microbial communities and the biogeochemical cycles they mediate in response to human-driven stressors. So in particular, I'll be talking about nutrient enrichment and some indirect effects of sea level rise. And so I'll start by admitting that I'm kind of leaning more on the biogeochemistry side today, but I'm always happy to chat microbes as well. So please, if there's any interest, feel free to take down my contact information, and I'd love to chat down the line. So before I start, I wanted to briefly describe my research. So I come from a pretty interdisciplinary background. I'm broadly interested in asking questions about the chemical and biological interactions that occur across scales and under different environmental conditions. And so what we're looking at here on the top is kind of one of my favorite pictures taken from a salt marsh pond. And it very clearly shows the spatial organization of microbes. So we can see purple sulfur bacteria as well as cyanobacteria. And in addition, we can see functions that they're carrying out. So here's like a little bubble from primary production, as well as decomposition of this leaf that's being carried out by heterotrophic bacteria. We can also see these types of interactions at a much larger scale. So in this case, on the bottom, a salt marsh, where there's interactions between the plants, the water, the atmosphere, and so on. And so ultimately, my research focuses on understanding microbes, the functions that they're carrying out, and how these things change along different environmental gradients, whether they be natural or human-driven. And so to put that more kind of concisely, my research theme is really understanding how coastal systems respond to perturbations and environmental change, and then trying to understand what this means for ecosystem function. And I address this question through a microbial lens. So really my question is throwing in the word microbes there instead, because they are so important in mediating many of, if not most or all of these biogeochemical cycles that we're interested in. And so that brings me to bringing up salt marshes. What better system than a salt marsh to address these questions? So salt marshes are one of the 3 blue carbon systems, in addition to mangroves and seagrasses, that are known to store carbon at very high rates. And so these are very highly productive, very dynamic coastal grassland systems, and they're situated right here between the land and the sea. And so they they're a really important filter between what happens on the land and what reaches the sea and affects the ocean. And because of this, they provide a really nice natural laboratory for understanding ecosystem function along different gradients, and really understanding the response to different dynamics that occur there. So in addition to just being a great laboratory, they're also just beautiful, as we can see in this picture here. And they provide a number of ecosystem services, such as habitat for economically and ecologically important species. They serve as an important storm surge barrier. So the plants facilitate sediment trapping and thus dampen wave action. They have a lot of cultural benefits. So fishing. When I was a graduate student at UMass Boston, we would bring out several first-year students out to salt marshes, just so they would have access to a natural environment. And one of the more important services they provide, in my opinion, is nutrient removal. So these salt marshes are hotspots for what we call denitrification. And this process is the reduction of nitrate, and it's a stepwise process to N2 gas, which effectively removes this nitrogen from the system to a largely biologically unavailable form of nitrogen besides those that are able to fix that nitrogen back into the system. And arguably my favorite is their ability to store carbon over long time periods. And that's because, we look at a salt marsh. So this is a salt marsh up from Massachusetts. We just see a very vast grassland with lots of primary production, green lush grass. And so that, in addition to the fact that below ground, we have very slow decomposition that occurs. And this is because the system goes anoxic very quickly. So decomposition slows down quite a bit. We see that salt marshes store carbon at rates that are much greater than other systems. And so what we're looking at here on the y-axis is carbon burial rate in grams of carbon per meter squared per year. So note that this is on a log scale. And then we're looking at, on the left, we have different terrestrial systems. And on the right, we have these blue carbon systems, salt marshes, mangroves, and seagrasses. And we can see that these salt marshes are storing carbon at rates that are much, orders of magnitude greater than that of terrestrial systems. We can also look at this plot that's showing the global averages of carbon storage for seagrasses, salt marshes, mangroves, and tropical forests. The navy blue shows the carbon that's being stored in the soil, and then the light blue is showing the carbon that's being stored as like plant biomass. And so what we can see here is that salt marshes, much of their carbon storage is as a result of this below ground soil carbon. Okay, so we'll kind of get back to that a little bit later. So despite the fact that these salt marshes provide a number of services, they also face a number of threats, much like any other coastal system. And so since the 1800s, we've seen a global loss of marshes. Of this paper, which is from 2006, cites 25%. Since then, it's much more than that even. This plot on the y-axis is showing the percent marsh loss for different New England states. Rhode Island, Massachusetts, New Hampshire, and Maine. And so, this just is showing kind of a more local example local to the marshes that I'll be showing you during this presentation. And so there are several things that contribute to this salt marsh loss, including invasive species, development, sea level rise, and nutrients. And today I'll be specifically talking about the last two. Okay. And so the outline for today is talking about salt marsh decomposition in response to nitrate, and then speaking very briefly to microbial-driven nitrogen cycling under elevated sulfide conditions, one of the indirect potential consequences of sea level rise. And so to start off with nutrients. So we know that human populations have increased, and so has the demand thus for nitrogen fertilizer. And so this map is showing the application of nitrogen fertilizer per hectare of cropland in 2017. And so very clearly we can see that there's just very high levels of application, right? So the darker blue colors are showing greater levels of application. And this fertilizer ultimately ends up somewhere near the coast. And so one question that we had continues to be, what does this increased nitrogen enrichment mean for salt marshes in particular? And so remember that salt marshes exhibit high rates of nitrogen removal through denitrification. And so typically they're thought to be fairly resilient to these increasing levels of nitrogen. But the question is there a tipping point at which these salt marshes can no longer sustain these high levels of nutrient enrichment? And so there have been a couple of really cool long-term experiments that have set out to kind of address this question. So the first one I'll be describing has gone on for over 40 years up in Cape Cod, Massachusetts, where they fertilized marsh plots with either ammonium or urea at 10 times, 30 times, or 90 times kind of the recommended level of fertilizer application. And what they observed was what you might expect to see in your garden if you were to add fertilizer. So this photo is, I believe, is a picture of this 90 times plot. Okay, so what they observed was elevated plant growth, green lush grass, and an increase in elevation. And so this would suggest that marshes are resilient to extremely high levels of nitrogen input, and that this is ultimately contributing to carbon storage by producing plant biomass and thus supporting sediment trapping. Okay, so that's kind of one take on the effect of nutrients on salt marshes. But there's another project that took place in northern Massachusetts called the TIDE project. And I believe this was about a 15-year project where they added 70 micromolars of nitrate in the form of dissolved nitrate. So what they would do is dissolve nitrate in like a big tank. It was named Frank the Tank, in fact. And this nitrate was applied to the salt marsh on the incoming tide. And so they observed something a little bit different from the last experiment. So this plot is showing the percent cover on the y-axis of Spartina above ground biomass. So Spartina alterniflora is one very abundant marsh grass. And on the x-axis is the year of enrichment. So at this point, the publication of this study, there's data up through 9 years. The holes are when fertilizer was not able to be applied. I believe this year is actually when there was a major storm and they didn't get the shipment of fertilizer to continue the experiment. But they persisted, which is great. And so the results was in contrast to the last study, right? So these open circles represent the reference marshes, so where no nitrate was added, and these closed diamonds were the enriched marshes where the 70 micromolar nitrate was added. And there's no difference in this above ground biomass in Spartina alterniflora percent cover. And so recall that the last study used ammonium and urea, and this one used nitrate, which is important because that's the primary form of nitrogen that reaches the coast. So that led us to ask, okay, so does the form of nitrogen matter? And so here's some results from a paper that just came out a couple days ago in Bioscience that suggests, spoiler alert, that the form of nitrogen does matter. And so what I'm showing you here is a meta-analysis of papers along the y-axis that have added nitrogen in different forms. So ammonium, ammonium nitrate, nitrate, and organic fertilizer, and measured the response of the plant community via just above ground biomass in this case. And so along the x-axis is the log response ratio. And what we can see is that for ammonium, ammonium nitrate, and the organic fertilizer, that there's a fairly positive response in biomass. However, when nitrate was added in these 4 studies, that response was much less, neutral, or even negative in some instances. And so here's the data, the same data summarized in a box plot where this dotted line is situated at 0. And so this makes it very clear that the log response ratio varies depending on the form of nitrogen that was used, and shows that nitrate exhibits a very different pattern from the rest. And so this might explain this deviation between the 2 experiments I just explained prior, one that added ammonium and then the other that added nitrate. And so the question is, if the plants aren't taking up this nitrate, well, who is? And so we have evidence that perhaps it's the microbial community. And so what we're looking at here is a PCOA plot from a paper published by a former lab mate, Patrick Kearns, back in 2016, while he was working on the TIDE project as a graduate student. And so I want to just take a second to explain this plot because I'll show another later in this talk. So each point represents a microbial community, or kind of the compositional makeup for a given sample. So points that are closer together have very similar microbial communities, and points that are further apart have very different microbial communities. And what we're looking at here is the active microbial community between reference marshes, and those are represented in these colder colors, so those that had no nitrate added to them, and then fertilized marshes in these hotter colors where, again, that 70 micromolar nitrate was added. And so we can clearly see at its simplest a shift in the microbial community between these 2 environmental conditions, the shift in when this fertilizer is added. Okay, so we know that the microbial community is responding to nitrate in some way. So what does that mean for function? So here's another paper that was published from the same project by Ketelcoop Jacobson and Ann Giblin in 2010. And they used a really cool method called push-pull, where they would add isotope into the marsh sediment using a pump and would let it incubate and then pull that sample back up. And so they found that microbial mediated respiration processes increased in response to nitrate. And so here we're showing on the y-axis micromoles of nitrogen in meters squared per hour for just fertilized and referenced marshes. And these different color bars represent different nitrate respiration processes, which I'll get to in a little bit. But the main takeaway here is that, we see a change in the active microbial community, as well as a change in the ecosystem function, right? So these microbes are changing the way they're using the nitrate. And so what we hadn't done yet, we know that this nitrate's being used, but what we hadn't done yet is understand a kind of more mechanistically this relationship between nitrate and decomposition. And so that led to this hypothesis, this question: are microbes competing with plants for nitrate to fuel anaerobic respiration? And so why have I made a big deal about nitrate in the first place? I just spent like 15 minutes giving you a whole narrative about the background of this project, besides it just being a super cool story. Nitrate can play a dual role in nature. So this, again, might explain why there is that fundamental difference between these experiments. So nitrate can either act as a nutrient, fueling primary production, right? And so this can contribute to organic matter storage, or it can serve as an electron acceptor in heterotrophic microbial respiration, which releases carbon dioxide. And this is important because the extent of salt marsh carbon storage largely depends on the balance between these two things. And although plants are very important and great, they're also complex. And so I'll admit that I focus mostly on the respiration part of it all. So I wanted to spend just a couple minutes talking about why nitrate is important for organic matter decomposition and microbes in general, so microbial respiration. So respiration, if we recall, is just a redox reaction involving the transfer of electrons, right? So in the case of aerobic respiration, what you and I are doing right now, hopefully you're breathing, glucose here is serving as the electron donor, and oxygen is serving as the electron acceptor, forming ATP, whoops, spoiling things, forming ATP and releasing carbon dioxide, right? Well, in salt marshes, oxygen is very rapidly used up within the first few millimeters of the sediment depth. And so other electrons like nitrate are used in its place. And nitrate is important because after oxygen is used up, it's the next most thermodynamically favorable electron acceptor. So in this table, we can see that when nitrate is used as the electron acceptor in a process called denitrification, it evolves much more free energy when compared to when sulfate is used in sulfate reduction. And this energy is represented in this delta G here. And so I mentioned these two specifically because they're the two most important electron acceptors in salt marsh microbial respiration. And so we can visualize this in kind of a simplified conceptual diagram where sediment depth is on the y-axis and some arbitrary concentration of electron acceptors on the x. Okay, so we can see that at the surface, oxygen is very rapidly used up, followed by nitrate, and then lastly, we have sulfate here. And sulfate is largely unlimited in salt marshes due to its high concentrations in seawater. We can also overlay organic matter quality onto this diagram with the more like reactive, recently deposited organic matter at the surface. And then as we move down the sediment column, this reactive organic matter is oxidized first because of its availability. And so we're left with less reactive organic matter at depth that some microbes can't use. And so this is what's stored over long time scales, upwards of thousands of years. So I want to stress again that this is, of course, a very simplified concept. There are tons of assumptions and exceptions here. But so through nitrate loading, right, I've been talking about nitrate loading that results from fertilizer and sewage, nitrate may be penetrating these sediments at depth. So it actually might make the profile look a little bit more like this. So as we increase nitrogen loading to coastal waters, what happens when we see nitrate at depth, especially when we see that plants aren't responding to this nitrate very strongly? And so if we're providing this more energetically favorable electron acceptor to organic matter that tends to be less reactive, where these sulfate reducers typically can't use it, what does this mean for organic matter decomposition and ultimately for long-term carbon storage? And so that leads me to some questions that I want to kind of address today. Does nitrate increase organic matter decomposition? So through the via microbial respiration, to be more specific. And does nitrate affect the microbial community? And I'll admit that when I was putting these questions together, I was, I was brand new to the microbial world, and really, like, what I should be doing is switching these questions, in my opinion. But so to address these questions, I went out to the salt marsh. This is a salt marsh again in Massachusetts, up in Massachusetts, and collected these cores from Spartina alterniflora, which is this grass right here along the creek edge. You can see kind of a clear transition between the grass types. So I collected it from this grass here at different depths. So 0 to 5 centimeters or shallow, 10 to 15 centimeters or mid, and 20 to 25 centimeters or deep. And this is kind of how I varied the electron donor. And then took those sediments, split them into two treatments, a plus nitrate treatment, where we added 500 micromole of 15N labeled nitrate, and then half of them just received filtered seawater, and this is considered the unamended treatment. So this is how we varied the electron acceptor. So I also want to point out that 500 micromoles is a very, very high concentration, but we did this on purpose because we wanted to make sure that nitrate was never limiting throughout this experiment. And I can answer more questions about that if you have any. I also want to point out that we added nitrate in the form of 15N, and this would allow us to track the fate of this nitrate into different processes. And so we took these sediments, we put them in these little flow-through reactors here. And these are really great because you can take, basically by measuring concentrations going into the reactor and coming out and tracking the flow rate, we're able to calculate different rates that occurred, whether it be respiration rates, DIC, denitrification, and so on. So from this, we measured biogeochemical rates. We looked at changes in organic matter characteristics throughout the experiment, which was 92 days long. And we also looked at shifts in the microbial community at the beginning and the end of the experiment by both looking at the 16S rRNA gene. And so 16S is this universal gene that all bacteria have that allow you to kind of identify the taxonomy of these bacteria, and also some metagenomics, which allows you to look at the range of genes that are present in these samples. So let's take a look finally at the first set of data. And so here we're looking at total microbial respiration measured as dissolved inorganic carbon. And so this is the combination of carbon dioxide, bicarbonate, and carbonic acid over time. And so what we have here in this plot is a DIC production in nanomoles per centimeters cubed per hour on the y-axis. And so to be clear, this is the change in DIC concentration between what's going in and what's coming out of the reactor, calculated as a rate, and it's corrected for flow properties. We have weeks after start on the x-axis. In yellow is that plus nitrate treatment, and in blue is the unamended treatment. And each point is the average and plus or minus standard error for each of those reactors. We had 3 reactors per treatment. And so for now, we're just looking at the shallow sediments as well. So if we think back to that simple conceptual diagram that I talked about, right? In the shallow sediments, we have exposure to more high-powered electron acceptors and more reactive carbon at the surface. So we'd expect a lot of DIC production and not necessarily much stimulation when we add nitrate. And that's sort of what we see here, where DIC production is high for both treatments. And although the addition of nitrate does stimulate DIC production a bit, it's not necessarily by much. But when we take a look at the mid and deep depths, the organic matter is becoming less reactive and perhaps less accessible to microbes. And so we see a greater stimulation of DIC production when we add that nitrate, okay? And so this is especially true in the deepest depths, where the addition of nitrate stimulates DIC production to nearly match that of the shallow and the mid depths. And so to get a more holistic view of the changes that are occurring here, we can integrate between these time points to look at cumulative DIC production over time. So essentially, it's like looking at all of the carbon that was decomposed and released in the form of DIC here throughout the experiment. So on the y-axis, we have total DIC production. So in micromoles per centimeters cubed for each depth. On the x-axis, again, yellow is the nitrate and blue is the unamended treatment. And so we can see a couple of things. One, that DIC production decreases with depth and hence reactivity. So ignoring even just the treatment altogether, right? But when we add nitrate, we can see that that stimulates decomposition between the two treatments, right? So here in the shallow sediments, nitrate and unamended, and so on. And so what we can kind of conclude from this is that this difference represents a pool of organic matter that microbes can access only under high nitrate conditions, a pool that would otherwise remain stable with sulfate alone as an electron acceptor. And this is interesting because it suggests that the proportion of organic matter that's traditionally considered unreactive can actually become available in response to nitrate. So it's kind of like a sliding scale depending on the environmental conditions. And of course, if you recall, I mentioned in the beginning this dual role of nitrate, right? So there could be an alternative explanation here, and that is that nitrate is simply stimulating greater microbial growth as a nutrient, and hence resulting in more respiration. And so that could be another explanation of what's going on here. But luckily, we added that nitrate in the form of 15N. And so this allowed us to track the fate of this 15N into either N2 gas, which is to quantify denitrification, or into ammonium, which would quantify what's called dissimilatory nitrate reduction to ammonium, or DNRA. And I'll speak more about these processes in a bit, but the important thing to remember is that these are respiration processes. And so here I'm showing a very similar graph to that of DIC, but in this case on the y-axis, we have the nitrate consumption in nanomoles per centimeters cubed per hour. The x-axis, sorry that the label isn't there, this is the weeks after start as well. The black line is this, again, this total nitrate consumption. So to be clear, that could either be respiratory uses, so they could be using it for denitrification or DNRA, or they could be using it for assimilatory processes, so for growth. So we can't, just by looking at changes in nitrate, we can't get at that. But then these bars, what they show is denitrification in white and DNRA in gray, so just respiration processes. And so this allows us to loosely kind of compare the two processes and get a better sense for how this nitrate's being used. And so what we can see for each of the depths, so that was shallow, mid, and deep, is an overall agreement between this total nitrate consumption and these respiratory processes, suggesting that to at least some extent, this increase in DIC can be attributed to respiration and not simply an increase in growth. We aren't just simply creating a bloom that's resulting in this increased DIC. So the next question is, well, what about the organic matter that these microbes are oxidizing under these elevated nitrate conditions? And so remember that organic matter is the electron donor in these heterotrophic processes. So in any given system, organic matter can consist of varying forms with varying complexity and accessibility, right? So more accessible or reactive organic matter traditionally has lots of nitrogen relative to carbon, resulting in a lower C to N ratio. So for example, we have phytoplankton or bacteria with a lower C to N ratio of like 5 to 8. And then we have less reactive organic matter here, such as Spartina, again, this marsh grass, that has a C to N ratio between 30 and 50. And so depending on what kind of organic matter the microbes oxidize, we might expect to see different byproducts from decomposition. And so to further examine this, we can calculate the ratio between the production of DIC and the production of ammonium. And from this, we can begin to infer what's being decomposed. So the higher the ratio, the more complex the organic matter that's been oxidized, okay? And so first, we can look at the unamended treatment, and we can see that across all depths, the DIC to ammonium ratio is relatively low and consistent. It also happens to coincide with the initial C to N ratio of the sediments, around 13 or so, indicated by this dotted line here. And this suggests that these microbes are essentially oxidizing the organic matter available to them, right? But when we add nitrate, we can see that this DIC to ammonium ratio increases across all depths, with a particularly noticeable difference in the deepest sediments, where this organic matter has a higher C to N and is typically less reactive and less accessible to the resident community, microbial community. Okay, so that gives us a better sense for what might be driving these changes in DIC. So I keep alluding to the microbial community and what they might be doing and driving these changes in biogeochemistry, so we can take a closer look at that. So first, I'm going to show you results from 16S rRNA gene sequencing. So again, this doesn't tell us anything about function. What this tells us is taxonomy, and it can give us some insight into the microbial community structure. Okay, so again, I'm showing you a PCOA plot where these points that are closer together are more similar. Okay, so the first thing is I want you to focus on just the shapes, which represents depth. So the circles are shallow, the triangles are mid depths, and the squares are deep depths. And we can see kind of a clear separation in the samples by depth. Right now, I'm just showing you the pre in green, which is samples that we took before the experiment started, and then unamended in blue. And when I put in nitrate here in yellow, we can see that in addition to these differences by depth, we also see that when we add nitrate, we're fundamentally shifting the microbial community quite a bit into something different. And so at this point, I can tell you taxonomy-wise, the different groups that are most relatively abundant in these treatments, where in the unamended treatment, we have microbes that belong to groups known to reduce sulfur. And I'll say that I'm using the language known to carry out these functions because, again, we can't say anything about the functions they're actually carrying out. We can only say things about bacteria that have been cultured and sequenced and rates have been measured. But when we add nitrate, we see bacteria that are enriched that are known to reduce nitrate and to degrade high molecular weight organic matter, and also, interestingly, to oxidize sulfur and actually carry out carbon fixation, which is really interesting. And so, so that gives us a taxonomic view. We also did some metagenomics, which gives us a better sense for the overall change in genes and gene abundance. And so first, this is just showing kind of just all the genes that are there. And the big takeaway here is on the right in green, we have the nitrate-enriched treatments. We see an enrichment in these nitrogen-related metabolisms, which isn't surprising, but it's really nice when you can see data that match up with your hypotheses pretty cleanly. And so to zoom in on those, for instance, under the nitrate-enriched treatments, we see an enrichment of gene abundance related to denitrification, DNRA, and ammonification, and then in the unamended treatment, interestingly, an increase in abundance of genes related to nitrogen fixation. And what I'm not going to talk about much today is we also saw a correlation between, I say nitrogen-related genes, but actually many genes and their biogeochemical cycles and rates that we measured as well. So that was really interesting to see. I am going to commit the cardinal sin of showing a figure and not talking about it, but I want to put in a plug for a colleague of mine, Joe Vinus, who's finishing his PhD in Jen Bowen's lab currently. And what he's doing is really the good work when it comes to understanding microbial communities. And he's taken a lot of these data and is creating metagenomic assembled genomes, so MAGs, so that we can look at the organismal level response to nitrate and really getting a better holistic view at the microbes that are being enriched. And so without, again, committing a sin, and I apologize, but basically for each of these lines, they represent a different assembled organism. And these are interesting genes that we're looking at. And so, many of them, for instance, possess the gene related to the genes related to denitrification, one really cool takeaway from his work, which we're very interested in pursuing further, is also many genes related to carbon fixation. And so one thing that we're hypothesizing now is that perhaps there's a lot more internal carbon cycling that's occurring here than we originally thought, and much more carbon fixation occurring in deep salt marsh sediments. Potentially, it would make sense that as carbon is being depleted from the system, that these microbes are finding ways to replenish that carbon through fixation. So there's a lot of interesting things coming from this story, so keep an eye out for that. And he's a rising star, so keep an eye out for him as well. And so to kind of revisit these questions, does nitrate increase organic matter decomposition through respiration? And the answer thus far is yes. And it does so by affecting changing and fundamentally changing the microbial community, as well as the metabolic potential of this microbial community. And so the key finding here is that nitrate can serve as an electron acceptor, not just as a nutrient, and can stimulate microbial respiration by accessing more complex forms of organic matter. And this is significant because as nitrate loading continues to coastal waters, this increased organic matter decomposition may diminish the capacity for long-term carbon storage. And so, of course, we know that several of these salt marshes along the coast are experiencing chronic long-term enrichment, right? So a next natural question we had was, okay, what about sites that have experienced this chronic enrichment, right? So the takeaway from this first story is that, sites that have experience high nitrate loading, their nitrate accessible pool becomes smaller. So does this have an effect on sites that have experienced long-term nitrate enrichment? Will they respond further to additional nitrate addition? And this has some implications for which sites we choose to manage and restore. And so what we did is we performed a very similar experiment, collecting samples mostly at deep depths from a reference marsh, so a marsh that had experienced very little nutrient enrichment, a marsh from the Tide project that experienced 13 years of nitrate enrichment and 70 micromoles, and then a 40-year enriched marsh that experienced between 10 and 1,000 micromole of nitrate enrichment. And so here we see increased nitrate enrichment both with intensity, amount, and duration. Okay, and so again, use that flow-through experiment. This experiment lasted for about 100 days. Very similar design where we added nitrate to half, and then the other half we kept unamended. And so I'll show you some results for that now. So again, we're taking a look at DIC production, which represents total respiration for each site. So again, DIC production in nanomoles per centimeters cubed per day on the y-axis, and in this case, to throw you off, to keep you on your toes, on the x-axis is the days after start. So based on the results from my first experiment, right, I expected nitrate to stimulate organic matter decomposition in reference sediments because this nitrate accessible organic matter pool is still available. And we see that here. So in this case, the blue is the nitrate addition, and then the gray, which will be the same throughout all the different sites, is the unamended treatment. And this is just for the reference site, so the site that has not experienced prior nitrate enrichment. But we also hypothesized then that sites that have experienced chronic nutrient enrichment, that this nitrate accessible organic matter pool would be smaller, because some or all of this pool would have already been oxidized during burial. And so when we look at these enriched sites, so this green, this is the 13-year enriched site. So the color is the nitrate treatment, the gray is the unamended treatment. And then here's the 40-year enriched site. What we see is that the effect size, the effect that this nitrate addition has, is much smaller when compared to the reference site, suggesting that perhaps this chronic enrichment, this organic matter that's been deposited, is affected by this chronic nitrate enrichment. And so we can take a closer look at this relationship by plotting total DIC production on the y-axis against what's called the aromatic to carboxylic ratio, which if you have any questions, I'm happy to talk more about it. But essentially, we got these data from Fourier transform infrared spectroscopy, which is a quick and rapid method that gives information on different chemical functional groups of a sediment sample. And so on the left here, so smaller ratios indicate more reactive organic matter, and larger ratios indicate less reactive organic matter. And so we can see here, again, blue is the reference, green is the 13-year enriched site, and orange is the 40-year enriched site. You can even ignore the shapes, which indicates the treatment. But we can see here is that respiration decreases with decreasing organic matter reactivity, supporting this hypothesis again that there is this less accessible organic matter that is remaining for microbial oxidation because most of it was already used as this organic matter is being buried. So there's a quick summary of that. And so I'd like to just end this narrative on the microbial community for this experiment. So here I'll be showing you a heat map of 46 different ASVs, which stands for amplicon sequence variants. So effectively a microbial species that were found to be significantly different between treatments. So the nitrate treatment versus the unamended treatment using a differential abundance analysis. Note here that I've removed the taxonomic information so as just not to overwhelm you. But if you're interested in the class level, that's indicated by the colored boxes. Each column here represents the relative abundance of these ASVs. So like the darker color in the box represents a greater relative abundance. And the 4 left columns are for the nitrate treatment, and the right columns represent the unamended treatment, right now just showing you the reference marsh. So where these sediments were initially collected from. So I just wanted to get you oriented to the graph first. So now I'll show you all 3 sites. So now we're including the 13-year site and the 40-year site. And so the main thing I want you to take away, I know that, getting good information from this in a short amount of time is very difficult, but I just want to tell you that in the unamended treatment, there are several taxa that are known to reduce sulfate. So different groups, right? So here that belong to the delta proteobacteria groups. And then in the plus nitrate treatment, many taxa that are, again, known to reduce nitrate, degrade high molecular weight compounds, and oxidize sulfur. So again, in this experiment, we found a lot of interesting evidence for carbon fixation. So definitely something we plan to pursue down the line. And so to kind of wrap this part of the story up, we found that chronic enrichment does affect further response to nitrate, in that this chronic nitrate enrichment may lead to less carbon storage overall, but the portion that is buried is less vulnerable to further decomposition. So after it's buried, the further nitrate exposure may not, lead to further decomposition, just because that organic matter that's there is just not, is less vulnerable to it. And so this is significant because this organic matter may be able to persist under high nutrient enrichment conditions for long periods of time. And what I didn't talk about today is we also looked at denitrification and DNRA between these 2 sites. And in the chronically enriched site, denitrification rates were much higher. So also built into this is that the microbial community is perhaps adapted to these high nitrate conditions, in that they act to remove a lot of that nitrate as N2 gas instead of retaining it in the system, which occurs with DNRA, which I'll get to in the next story. So the conclusion here is that microbes are using this nitrate to fuel anaerobic respiration and not necessarily just to support nutrient requirements. And this results in greater organic matter decomposition. And so this may explain some of the differences that we noticed in the 2 experiments I explained in the beginning of the talk, and is also significant because more organic matter decomposition not only contributes to CO2, to greenhouse gases, right? But it's the amount of sediment that it creates also dictates whether or not salt marshes can survive sea level rise. And actually, one of the major outcomes of that tide project that I explained was that they found that in the fertilized sites, these marshes were beginning to crumble. and slide into the creek bank, and just large clumps of sediment that represent thousands of years of carbon storage were just disappearing. And they think this is partially due to the fact that these below ground respiration processes were being stimulated, as opposed to the traditional thought that plants would simply respond and actually lead to greater carbon storage. And so I want to just end on a really quick story talking about microbial nitrogen cycling under elevated sulfide conditions. It's very short, I promise. And so we know that sea levels have been rising, as suggested by this plot here, based on the 2018 IPCC report. And given that salt marshes reside on the coast, it shouldn't be surprising that sea level rise has some major consequences for salt marsh health and survival, which I just alluded to. And one potential indirect effect is the increased prevalence of sulfate reduction. So, right, we talked about nitrate reduction. So in this case, sulfate reduction, sulfate is being used as the electron acceptor, and it produces concentrations of hydrogen sulfide. And sulfide can be toxic to plants. It also is important to the microbial community by altering biogeochemical cycling. Remember, I also mentioned in the last work that many of these microbes were capable of oxidizing sulfur. So the sulfur cycle is very complicated, but and I don't claim to be an expert at all, but can play plays a really important role in salt marshes. And so recall I mentioned that there are different forms of nitrate respiration, and I've talked about both of them, but very, very briefly. And both of these use nitrate and organic matter. And so the one that I've really focused on thus far is denitrification. And so this results in the removal of nitrogen from the system as N2 gas. But there's also a competing process, again, this what is called dissimilatory nitrate reduction to ammonium, which I'll refer to here as DNRA. And in this process, rather than this nitrogen being removed, it's actually retained in the system and can be taken up by plants. And so there are several different conditions like nitrate concentrations or organic matter quality that favor one process over the other. But high concentrations of sulfide are under question, and they've been known to inhibit the final steps of denitrification and also to promote chemolithoautotrophy, so an autotrophic process using chemical energy. But generally, there's much less known about how sulfide affects this partitioning. And understanding what process occurs is really important and has important implications for water quality, as you can imagine, where, again, one process removes this nitrogen and the other retains this nitrogen in the system. And so we set out to ask how sulfide influences partitioning between denitrification and DNRA. And so this was in collaboration with a postdoc, Annie Murphy here, and we were led by a very, one of my favorite experiences, led by an undergraduate student, an REU student, Ross Ackerman, who collected sediments from the marsh and conducted anoxic batch incubations using sulfate-free artificial seawater. And he also added 70 micromolars of 15 labeled nitrate, again, so we can track the fate of this labeled nitrate, and varied the concentrations of sulfide from 0 to 1,000 micromolar of sulfide. So he again looked at the labeled N2 gas, which gives us information on denitrification, and then labeled ammonium, which gives us information on DNRA using a membrane inlet mass spectrometer. And so I'll show you a few graphs now that's showing these results. So first, we're looking at the nanomoles of grams per wet weight per hour for denitrification. And along the x-axis is the concentrations of sulfide used in this experiment. So the first thing we can see is that as sulfide concentrations increase, these denitrification rates decrease, with a significant difference occurring here at the highest concentration of sulfide. We realized after the fact, as we always do, that we probably should have used much higher concentrations of sulfide here, but luckily we still saw some trends. As opposed to, again, so denitrification is removing this N2 gas from the system, as opposed to DNRA, again, which retains the nitrogen in the system as ammonium. We saw inconsistent, but generally, an increase in DNRA in this 100 micromolar concentration of sulfide, and some changes in the higher concentrations. So to get a cleaner look at this, What we did is we calculated the ratio of DNRA to denitrification for each of these treatments as well. So this dotted line, anything above it represents more DNRA, and anything below it represents more denitrification. And what we can see here is that under the highest concentration of sulfides, we see a greater contribution of DNRA to this nitrate respiration process as opposed to lower concentrations. And so the takeaway here is that sulfide resulted in this greater contribution of DNRA relative to nitrification, which suggests that there's greater accumulation of this ammonium in the system, which, at a greater scale, has very important implications, again, for water quality. It also shows that there's a really tight coupling between nitrogen and sulfur cycles. So what I, admitted to in the beginning is I leaned a lot more towards the biogeochemistry. We also sequenced the 16S rRNA gene. We looked at both the total and active microbial community here as well, and found that throughout this incubation, the microbial community significantly shifted as well, favoring groups that were known to reduce sulfate, oxidize sulfur, really push towards the community that was able to take advantage of this increased sulfide concentrations. And we saw a decrease in nitrogen-related or taxa known to carry out nitrogen-related processes. And so bringing it all together, right? Salt marshes perform so many critical ecosystem services, particularly the service of carbon storage as well as nitrogen cycling. And as we put forth effort to restore these systems, we need to keep in mind the effect that things like nitrogen, specifically in the form of nitrate, and sea level rise has on marsh microbial communities and the biogeochemical cycles that they mediate. And so hopefully this work has provided a more mechanistic understanding of that. And at the very least, I hope I've convinced you that taking this holistic interdisciplinary approach can help to address some of these questions. And so with that, I wanted to stop to leave time for questions and acknowledge the so many people that have contributed to my development over time, great advisors and co-authors, as well as my funding. And with that, I'll take any questions.
Awesome, thank you for that great presentation. So normally we like to open up questions to students first. So students, if you guys have questions, go ahead and raise your hand and I'll call on you. So how can I keep this slide up, but then also see everybody? I think you'll only be able to see us in in the bar below. Okay. If you want to still keep the slide up, is my understanding. Maybe what I'll do is, yeah, we could go ahead and people can ask questions and I'll figure this out. Okay, I'll do that. Okay, I see Kirsten has a question. Go ahead.
Hi, Ashley. Thanks. That was really interesting. So in the beginning, you said that the amount of carbon storage in marshes is similar among mangroves and seagrasses. And I know that these blue carbon ecosystems are often lumped together. So do you think that nitrate enrichment will have the same effect on microbial activity and community composition and carbon storage in mangroves and seagrasses?
I think that's a great question. And I would venture to guess that the response would be similar just because of the similar processes that are carried out among these systems, but we really don't know. We really have a very, very small handle on the microbes within these communities as well as how they might respond. So I think that's a great question and something that I'm hoping to pursue as I continue my work here at Eckerd.
Okay, thanks.
Yeah.
Ashley, I typed your contact info into the chat, so if you wanted to see everybody.
Yeah, that would be great, because I don't even know if you can see me because the sun's going down and it's dark. Okay, great. Sure. All right. Okay. Any other student questions? I'll ask mine as we wait. But I saw that in your PCOA plots after you had added
the nitrate and stuff, you saw that community shift of like those mid and deep regions, and those seem to really overlap with each other. And though like you did see that shallow region seem to start to kind of overlap with them, it wasn't like as strongly. Did you have like any hypothesis for what might be going? Is there like, do you think there's like a base microbial community like in the shallow that's already like used to this, that's set up? I don't know.
Yeah, no, that's a great question. I think there's a couple things at play that I believe may be driving those patterns. One, that nitrate does occur a bit more, in the surface. So it is present in the surface. There's also a process called nitrification, which leads to the production of nitrate, but that only occurs under oxic conditions. And so that's one thing that may be separating the shallow sediments from the mid and deep. The other thing is, what I didn't mention is the shallow sediments, we designed it such that those were collected kind of from the depth where there is the rooting zone still, and the mid and deeper depths are kind of beyond that rooting zone. And so that also plays a really big role in structuring the microbial communities. As we know, there's a really tight coupling between plants and microbes as well. So that could also be driving the response there.
Okay, great. That makes sense. Okay, so I'll open up to everybody else, and I see Maya already had her hand up, so go ahead, Maya.
Hi, Maya.
Hey, Ashley, thanks. That was fascinating. So I was trying to put my hand down, and I can't multitask at the same time. So I'm wondering about the timing of the experiment, and I'm trying to figure out. So, like in your Unamended, where you did like the normal samples versus the kind of ones habituated with the nitrate. Like you see the kind of biggest peak at like 60 days, but then kind of by 80 days or the end of your time point, it's gone back to the same as the Unamended. So is it like that they kind of pulse and react, and then that's over, or what do you think's going on timing wise?
Yeah, so let me see how easy it is for me to just put this back up real quick. No, that's a great question. And so what we believe, so unfortunately, we weren't able to collect microbial samples at each time point, which would have been great. So a lot is left up to speculation. There's a couple things. One, it could be that, first off, that the microbes just take a while to get going and to get accustomed to their new environment, right? So this was not in situ, this was a laboratory incubation. So to some extent, there's some adaptation that needs to take place. The other thing is, after they become accustomed and they, are able to figure out a way to use that nitrate, what we think is maybe they're using the organic matter that's available to them, but then over time, that organic matter starts to decrease again, that's available. And so if we were to continue this experiment for another 100 days or so, which we definitely avoided for the sake of my mental health, we would expect these two treatments to eventually converge, with the idea that whatever organic matter that these microbes were able to oxidize would eventually be gone. The last thing that's coming out of more of the metagenomic MAG analyses is, well, is there a switch potentially, which we can't really answer, unfortunately, but is there a switch between heterotrophic respiration and then more carbon fixation occurring, such that maybe the same amount of decomposition is occurring, but we're not actually picking it up in the DIC because it's being rapidly fixed again. So a lot more of that internal carbon cycling occurring than we would expect. We can't answer that question, but it's a kind of a cool concept that we're trying to set out to address again. So hopefully that answers that question.
Definitely, thank you.
Yeah, thanks.
Okay, so I saw Steve had his hand up next, so go ahead, Steve.
Thank you, Natalie. Thank you, Ashley. Great talk, and congratulations on your new job at Eckerd. Eckerd has a lot of really good sedimentologists over there, and so I'm sure you've reached a home there that you've got a lot of expertise and friendly faces. So that's great.
Absolutely.
I wanted to ask you a very practical question,
Sure.
and that is, back 50 years ago, there was an oil spill in Wild Harbor, which I think is probably one of your study sites, or at least close to it, right? And John Farrington and Chris Reddy at Hui have been monitoring that for 50 years, and they found that after the oil got to a certain depth in the sediment, it stopped weathering, right? And they attributed that mostly to the fact that it was reaching the anoxic zone. And your hypothesis is that basically it may be nutrient limited with nitrogen. So the practical question is this a remediation strategy by, adding, excess nitrogen to see if we can actually degrade, the oil that might be, below the oxygen zone?
That's an interesting philosophical question. I think the question is what your goals are, right? So, it's one of those things to keep in mind. Are you doing more good than harm, or doing more harm than good? And so, it could potentially be providing more, changing the thermodynamic landscape and allowing for more of this oil to be degraded. But at the same time, it could also be leading to more carbon respiration. So you're losing a lot of that stored carbon as well. And so may potentially
the
work.
oil stored carbon, so I don't know. We have the same problem from Deepwater Horizon that's trapped in the Louisiana marshes doing the same thing. And there really is no remediation technology, that can be applied, right?
Yeah. Yeah, no, absolutely. No, it's an intriguing question for sure.
I think you ought to experiment on that.
Yeah, that sounds like a great experiment. Yeah.
Thank you.
Thanks. Thanks for the idea.
Okay, Christopher, go ahead.
I don't know can you see me?
Yeah.
All right, nice talk. Very good talk.
Thank you.
So I'm a research geologist over at the USGS, and so I'm a coastal geologist. So when you were talking, I was thinking you presented a lot about the organic substrate on which constitutes these marshes, but I also think about how Florida is kind of on a gradient in terms of the inorganic type of sediment that may be in a marsh. Right. And so I was wondering if you might comment or speculate, I don't know if you have much data on it, but how the inorganic substrate, so clays versus carbonates, may influence some of the biogeochemistry that you're seeing.
No, great question. And I failed to mention that these marshes up in Massachusetts that I used for these experiments are very organic rich compared to the marshes down down further south. And so what I would hypothesize is that their response to these kinds of experiments would probably be less with this higher inorganic makeup of the sediment. But there's, it's hard. There's great value in doing these, lab incubations because it allows you to isolate, these processes from a lot of other things. Like I mentioned, it isolates it from the contribution of plants, right? The other thing at play when thinking about carbon storage and accretion in these salt marshes is like sediment accumulation and, especially inorganic sediment accumulation. So I guess to not necessarily answer your question, but I'll admit I don't have data on marshes that are more inorganic, but that's definitely something that I'd be interested in pursuing as well. And I have a collaborator at Sapelo Island in Georgia where we're trying to look into this and to really parse apart the differences that the organic matter versus inorganic matter content makes in decomposition under different environmental conditions. So no, great question. Thank you.
Would you expect, just kind of a follow-on, would you expect the microbial community to differ considerably between the two marshes?
I would expect so, yeah. I mean, it's a good question. We don't even have, there's so few microbial data on marsh sediments and mangroves too, and seagrasses, but that we don't even really have a good feeling for geographical variation. And marsh to marsh, and even just along a depth gradient and things like that. I would expect it to vary, especially because of the importance organic matter plays in driving microbial respiration. And so I'll leave it. Again, that's another question that we're hoping to pursue when we kind of compare these organic rich versus inorganic rich marshes.
Great, thanks.
Yeah, thanks.
Okay, Isabel, go ahead and ask your question.
Hello, great presentation. I was just wondering, because plant microbial interactions are key for decomposition and as well competition in coastal system. So have you considered to look at the plants, which nutrients are limiting these plants? Because a lot of times, at least in mangroves, depends on the nutrient that is limiting the mangrove tree. You will have a different response of the system, as well as the microbes and the plant itself to the nutrients that you're adding. For example, if a plant is limited by nitrogen, and you add any type of, especially nitrate or ammonium to the system, then you increase decomposition because the plant is not using that nutrient and the microbes just get crazy. But if you add phosphate and they are limited by phosphate, actually you can restore the forest because then you get all these carbon from the plants store on the soil, and then you see a growth in the forest. So I was just wondering with salt marshes, did you expect that it's possible to see a similar trend?
Yeah, no, great question. I mean, we admittedly don't do any really touch phosphate when it comes to these kinds of experiments. Mm-hmm. We also don't see a lot of evidence for changes in phosphate related metabolisms when we look at the metagenomes of the samples we've looked at thus far. And I think I agree that the plants are a very critical component to the overall processes that occur in the marsh. And I think what was driving this initial experiment was the fact that when we looked at the different forms of nitrogen that were added, right, like ammonium versus nitrate, that when we added nitrate, that the plants weren't responding at all. Like they weren't, we were shocked to see. At first, we're like, this 13-year experiment has failed because we saw no response in the above ground biomass in the plants. And so that led us to then hypothesize, okay, something must be taking up this nitrate. It must be going somewhere, because it also, what I didn't mention is we also did a marsh-wide isotope addition where we tried to track the fate of this isotope that we added to the entire marsh. And it also wasn't being exported. So it was going somewhere. This missing nitrogen was going somewhere, nitrate. And so that's what led us to really dig into the effect of nitrate on the microbes and stuff. But I think, these kinds of experiments, not this one because of the high concentrations of nitrate we used, but these forms of experiments they really lend themselves well to modeling, I think. And so I think that's like a really good next step with kind of carrying these out to provide kinetic parameters for these models that can do a better job of thus then predicting what's happening at the ecosystem scale, while taking in both these, respiration as well as the plant communities, as well as the organic matter makeup into account.
Mm-hmm. So you're planning to do some of those experiments with like seagrasses down here, or you're going to continue your experiments up there?
Oh no, I definitely want to try and move into the seagrass mangrove world. So hopefully you all can help me with that.
Oh, that would be awesome. Yeah, I work on looking at the mangrove trees itself and how they can affect the microbes. So it's kind of working at the same, but looking at a different perspective, and it's very interesting what you can find. Yeah. Awesome.
Yeah, absolutely. Yeah. Great. Thank you.
No, thank you.
Okay, I don't see any more hands raised, but if anybody has some questions, go ahead and ask them.
And then
I know it's Friday. I know it's Friday afternoon, so.
I was going to say, usually we will end with inviting everyone to unmute and give you a round of applause. But before that, I just want to remind you all about the Toast to Graduates at 4:45. So hop on over there. And yeah, Ashley, huge thank you for giving a seminar. Congrats on surviving your first semester as an assistant professor in a pandemic. An excellent talk. If everybody wants
to. Thank you so much. Yeah, thank you so much for coming.
Thank you so much, Ashley.
Thank you. I hope everyone has a wonderful holiday.
Thank you.
All right. Take care.
Thank you.
Hey, Ashley, are you still there?
I am.
I have another question, but I don't make everyone listen to it.
Yeah, no, totally.
So that was awesome, and I'm super excited. We should absolutely talk whenever you have bandwidth about.
Yeah,
We have a ton of freshwater springs metagenomes that we're not even doing anything with now, but like they really need to be looked at with somebody who knows something about biogeochemistry, which is not me. But also, like, I thought it was cool that so many of your mags had genes for, if I read it all quickly and right, like had genes for nitrogen, for denitrification, and for like various sulfate